All right, chapter 9. Now, when we look at chapter 9 material, it's all about the muscular system and how it functions. Keep in mind that we have three types of muscles: smooth, cardiac, as well as skeletal muscles. Now, cardiac and smooth we'll talk a lot more about later on. And in general, we don't talk as much about smooth muscle as we do the other two type of muscles. So this is the main focus on skeletal muscles. So when we look at the muscular physiology, understand that when we're talking about muscles, like your biceps muscle, biceps brachii muscle, which we will kind of go through again, right, next week as we kind of discuss all the different parts of the muscles and all the different muscles you need to know for the next lab practical. Now, when we look at a big muscle group, like your biceps, your quadriceps, your hamstring muscles, well, those muscles are a muscle group, clusters of muscle cells. Now, here's one cluster right here that we call a fascicle. So this is what we look like if we take a cut of your bicep and look at one cluster or one fascicle of a muscle. So you have all of these muscle fibers, individual muscle fibers, and each muscle fibers are going to have proteins inside that allows it to interact with other muscles, right? That allows it to react with other proteins. Let me phrase that. And as it interacts with other proteins, it allows those proteins then to move, to slide past each other, and then allows them to eventually, right, this is just going to turn on in a second, eventually once it slides past each other, to shorten. Now when we look at muscle systems itself, for some reason it's skipping, so go back here to not full screen mode, I guess. When we're looking at the actual system itself, come back all the way around. These are the general properties. All muscles have the ability to contract, which means that they have the ability to shorten and then pull something with force. Keep in mind, contraction, and we always refer when you're working out, right? You ask somebody to flex. Well, flexion and contraction are slightly different. Flexion can be caused by a contraction. Flexion is an angle between the two bones you're moving. If the angle from a straight line of 180 degrees with your arm straight down in like an anatomical position, and then you move your arm up, that's flexion. Now, you can also contract your muscle and cause extension. So once you start flexing, you extend your elbow again. Contraction then causes an extension effect. So contraction and flexion doesn't always mean the same thing, because contraction on different muscle groups can cause the opposite effect and give you extension. So contraction is a word that we'll be using a lot more than either one of those two. So when we ask somebody to contract the muscle, well, sometimes contraction of the muscle will cause you to have flexion, sometimes extension, depending on the muscle that you will start to contract. Keep in mind that when muscles contract, though, they always shorten. So if you think about where the muscle starts and where it ends, you can kind of figure out the actual movement that it makes. So your bicep starts from your scapula, goes to your radius, and the anterior part of your arm. So once you start contracting it, it makes sense then the actual arm and the humerus and radius will start, the actual radius and ulna will start to flex. As it starts to flex, the angle of your elbow joint starts becoming less than 180. Excitability, capacity of muscle to respond to a stimulus. All skeletal muscles require nerves. But once you have a motor neuron that stimulates the muscle, they need to be able to excite throughout the extent of the muscle group. All right, so this is our way of turning on the cell. Extensibility. Muscle cells can be stretched to its normal resting length and beyond. You've seen people doing yoga, and usually women are eminently more flexible than men. Well, that is the extensibility of the muscle, right? So extensibility of muscle, you can stretch it beyond its normal resting length and beyond. And in some people that are like really hyper flexible, you'll have increased extensibility as a result of this. Last one, elasticity. Ability of muscle to recoil to its original resting length after being stretched. Now, the best example of this right here, now keep in mind, this is general properties of all muscles. So smooth muscle, best example right here. When the smooth muscle of your bladder is stretched out. So your bladder is basically one organ where the vast compartment of it, the vast cells that make up that organ are smooth muscle cells. And we know for a fact that it can stretch a tremendous amount. The more it stretches, the greater filling, the more urge to urinate. And what happens? That extensibility allows you then to wait a little bit. Even though you have this urge to urinate, you don't want to just urinate in front of everybody. So you have time to wait a bit until you can go to the bathroom and then relieve yourself. Once you relieve yourself, you get rid of that urine, and now it goes back to normal positioning. So extensibility and elasticity are really a good example seen in smooth muscle. We see it in skeletal muscle and cardiac muscle, but you're not going to see as much like extensibility in skeletal and cardiac because we're not going to really hold that, right? Not going to be able to stretch it, and we won't need to stretch it from a resting position as much. Now, I just kind of mentioned skeletal, smooth, and cardiac muscles as the three types of muscles. Some of them, especially skeletal muscles, are always voluntary, but there are muscles that are voluntary up to a point. One of the muscles that are the most important single muscle in our body, your diaphragm. Diaphragm is a skeletal muscle, but it's voluntary because it's skeletal in nature. The thing is, there's a limit to how voluntary it becomes. You've seen I know, Nicole, that you have kids. I have kids. My kids are old, right? Not old, but they're 16 and 21. Well, with kids, when they're younger, if they don't get a toy, they seem to throw a tantrum, right? So they would hold their breaths. And usually holding their breath is a voluntary movement. Now, you wait them out, they will pass out, and then they will resume breathing again. In other words, there's a limit to how hard you try to control your breathing. Usually kids are not that stubborn. Once they kind of feel uncomfortable holding their breath, then they stimulate the diaphragm and they start to respire again. So there's a limit to that voluntary nature right here. Respiratory movements, other type of body movements. Smooth muscles found in the walls of hollow organs. Now, hollow organs doesn't mean that they're always hollow. Hollow organs mean that it's nothing really solid inside of it. You can have blood inside of that organ. You can have urine inside that organ, or you can even have a baby inside that organ, or feces inside that organ. So all of those are hollow organs until there's something in there. Your uterus, very, very hollow, unless there's a baby inside. Then it's very, very not hollow, right? Very dense. So these walls of hollow organs, if you think about it, we need a muscle that will expand quickly. And then once you relieve yourself of that condition, like urination, defecation, it can then recoil to its normal resting length. Not only that, we want an organ where you really don't have to think about controlling all the time. Can you imagine how boring your life would be if you have to keep thinking about activating the smooth muscles of your GI tract as you are digesting and absorbing food for 4 or 5 hours at a time? And you have to voluntarily cause this effect. Well, yeah, we don't want that kind of very boring thing to do. So smooth muscles are found there. Smooth muscles doesn't require active control. It's involuntary. It can be controlled by the endocrine and the autonomic nervous systems. Now, in some areas, it's autorhythmic. They have pacemaker cells there that stimulate. And once you stimulate it, you have a wave of action potentials that will cause the movement of food matter to go from one region of your esophagus down to the stomach to the rest of the GI tract. Again, you don't want to have active control for that long of a time. Where else do we see it? In the eyes, allowing us to dilate and constrict pupils. In the hollow organs, we see it propelling urine, holding urine, and then regulates blood flow. So your blood vessels are made out of smooth muscle, so you can control your blood pressure. How do we control it? By opening, vasodilating, or closing the capillaries, as well as controlling the arterioles. So then we can regulate blood flow and blood pressure. Now, we don't think about it in general in terms of blood flow, but your blood vessels is one continuous hollow organ that unless blood is in it, it's going to be hollow. Cardiac muscle, the heart. Cardiac muscle can also be known as myocardium, cardiac myocytes. So in terms of these muscles, the heart is the main pump, allowing for a major source of movement of blood. They're always autorhythmic, always controlled involuntarily by both the autonomic nervous system and the endocrine system. Now, autonomic nervous system we'll talk about later, but that's your fight or flight responses. Also your rest and digest. So it's sympathetic and parasympathetic. There's always a balance between the two. So as you're sitting here in my lecture, getting bored already, right? You have more parasympathetic activation. And then as you're about to go work out, you have more sympathetic. So this is a good picture just to review the differences. Skeletal muscles, multinucleated, smooth muscle, single uninucleated kind of cells. Cardiac muscle, single centrally located nucleus. And you can kind of go through all this, try to, this is a good slide just to maybe even print by itself so you can review it and write on it, because they have so many things that they discuss right here about the different functions. Now, I say people generally do not control smooth muscle, but there are people monks in the Himalayas that have somehow actively controlled their smooth muscle. And by actively controlling their smooth muscle, they can vasodilate the blood vessels somehow when they should be vasoconstricting. And they did a study, and they actually showed it on TV once that I saw on Nat Geo. And they had the monks of the Himalayas, and they had these, monks. And what they did is to prove that they're really good at concentrating and to get into that trance, right? What they would do is have monks go up as high as they can where it's really cold. In the temples, they open the windows and open the door. So then it's really cold inside. And then they disrobe the top of their robes, which is just a robe anyway, so it's not much. And then their job is to, number one, all of them has a wet towel over their shoulder. And their main purpose is to somehow, through that trance-like state, their main purpose is to somehow create enough body heat to dry the wet, cold washcloth. And that's covering their shoulders. Remember, it is freezing cold, and the only thing they can do is just meditate. As they meditate, they somehow are able to control the blood flow to the skin. So then, and they actually showed it on the actual FLIR or infrared camera. They actually showed that the body temperature in the upper chest shoulder area increased more than even in the abdomen. Somehow they're able to control the smooth muscles that made up the blood vessels leading to the skin of the torso, and they're able to relax them, dilate them, so more warm blood course through, allowing them then to generate heat. So it's kind of cool. That does happen. Most of us can't do that. It takes a lot of meditation. Now, scientifically, there's another word for meditation. We don't like using words like that. It is called biofeedback. But we're talking about the same thing, where you're focusing on breathing, trying to put your mind at ease. Now, when we look at the skeletal muscle structure of a cell, number one, a skeletal muscle fiber is the same as a skeletal muscle cell. So those words can be used interchangeably. These skeletal muscle fibers or cells are long, cylindrical, with multinucleated cells that can be pretty, I mean, we're talking about long, most cells are microscopic, right? These cells right here that are the skeletal muscle fibers can be up to almost 30, 40 centimeters, which is pretty long when you consider normal typical cells are microscopic. So these are pretty long cells, and when we look on the actual cell under a microscope, you see the banding, light, dark, light, dark, light, dark banding. These are the striations. Now, in big muscles, like your quadriceps, each of the cells for the quadriceps are going to be thicker and longer. In smaller muscles, like your thumb and your fingers, each of the cells are going to be shorter and smaller, right? There's always a balance that we're going to talk about in our body. In bones, we talk about the balance between strength and flexibility, right? Here, and even in joints, strength and flexibility. In muscles, it's about strength and precision. So the more strength you need, like the way muscles need to contract so then you can sit still and watch a movie, right? That's a couple-hour movie. So you need muscle contraction of your erector spinae. That's more strength than precision. So those muscles that make up your erector spinae need to be bigger. They need to be stronger than the muscles of your fingers that usually don't do much in terms of lifting something with a lot of strength. They develop from individual myoblasts that then fuse together. The number of skeletal muscles remain constant throughout life. The big thing is understanding that when we work out and we get stronger, when we work out and our muscles grow, it's not that you add more muscles. It's that each of these muscles, skeletal muscles right here, get damaged. Then once they get damaged, they heal. During the process of healing, you activate protein synthesis. So then you have more proteins inside each skeletal muscle fibers. More proteins, larger cell. More proteins, also stronger cell. Next. Now, what I want you guys to understand is that skeletal muscle cells, even though they look different, are very similar to a normal cell. In a normal cell, we have the plasma membrane. Over here in the skeletal muscle cell, we see the plasma membrane, right? Covers the outside. So this is one muscle cell. And this is a cut of a couple muscle cells of a fascicle. Again, in the biceps muscle, it's a muscle group. And what we're going to see is it's not made out of one muscle cell. It's not strong enough, an individual muscle cell. So instead is one muscle cell clustered around another 100 muscle cells we call a fascicle. And then you have lots of fascicles to make up the actual muscle group. So a lot of individual muscle cells are clustered together to form a fascicle. Then you have lots of fascicles to form the actual biceps group. So now we're looking at just one muscle from one fascicle. And what do we see? Just like a normal cell, plasma membrane. Just like a normal cell, organelles. Just like a normal cell, some kind of endoplasmic reticulum system, right? We also see sarcoplasm or cytoplasm, like a normal cell. So it's very similar to a normal cell, except that there's some differences that allows it to do its job. So these muscle cells are long, cylindrical, and multinucleated. The plasma membrane is called the sarcolemma. Now, get used to using this root right here, sarc. Sarc is going to be specific for muscle fibers. So sarcolemma, the muscle cell plasma membrane. Whoops, right? So the muscle cell's plasma membrane is a sarcolemma right here. Now with the sarcolemma, the main purpose of it is to transmit action potentials. It's going to have lots of voltage-gated sodium, lots of voltage-gated potassium channels, and also other sodium glucose receptors as well, things like that allows fueling. Now, with the sarcolemma, if you look really closely at the sarcolemma of a skeletal muscle, right? So imagine you have a skeletal muscle cell in front of you, right? And I've blown it up so you can see it without a microscope. When we're looking at the skeletal muscle cell in front of us, you see the outer membrane, sarcolemma, and then if you look close enough, every now and then you see little holes on the inside of the membrane. The holes are pockets of the plasma membrane that's been pushed inwards right here. And they form tunnels. And those tunnels are actually part of the plasma membrane that's been embedded and pushed interiorly. So whatever happens at the plasma membrane gets carried into these tunnels. Those tunnels form a triad called, the triad is the tunneling of the plasma membrane. That itself is called the transverse tubules. So the transverse tubules will receive action potentials from the cell membrane surface and carry it deep into the cell. As it carries it deep into the cell, it is surrounded on each side by what we call terminal cisterns. These are large holding tanks for calcium. Now, a cistern, and we do see cisterns all the time, especially in the old world, right? In Greece, in Turkey, right? In the Middle East, where it's not raining a lot, what happens is when there is rain, they need to be able to store as much of that rainwater as possible. So in Turkey, you'll see that there are huge underground cisterns built so that when there's rain, water can collect in these underground cisterns for later use. A cistern then stores that water. Here, we're going to see terminal cisterns as part of this web-like reticular system that surrounds each of the protein that are found inside the skeletal muscle fiber. Right, now what else? We'll come back to transverse tubules and the triad in a second. What else is this? Sarcoplasm. And this is very important. I'm highlighting some of this. It should be highlighted before, but that's okay. And I'll post this up. The sarcoplasm is a muscle cell cytoplasm. In terms of the sarcoplasm, there's a couple of differences than a regular cell. Number one, the main purpose of this cell is to shorten and contract with strength. It makes sense, then we're going to have clusters of proteins that are going to allow that contraction. Since its main purpose is to contract with strength, right? The whole internal aspect of the cell is filled with these proteins that are arranged in a system we call myofibrils. So here's one myofibril, another. Again, right? Inside each cell, lots of myofibrils where the proteins that we're going to kind of understand are meant to contract, where the proteins will be arranged. You have lots of myofibrils per skeletal muscle. The more you work out, the more myofibrils you form. The more myofibrils, the bigger the cell gets internally, the more the muscle grows. What else do we see besides these huge amount of proteins that are then organized into myofibrils? You see in the sarcoplasm, lots of mitochondria. You see the nucleus. Since the purpose is to contract with strength, you want to store and hold as much or as many myofibrils as possible. So much so that you push the nucleus to the side, making sure that it's out of the way. So you have the nucleus, there's a lot of them, and they're peripherally located. What else? Inside, in the sarcoplasm, we're gonna have lots of myoglobin, red-colored oxygen-binding protein. Not only will we see myoglobin, but we'll also see lots of glycogen. So we have lots of glycogen, lots of myoglobin. Perfect. It allows the muscle to contract for a few minutes at a time before it requires fuel from the bloodstream. Remember, when we are working out, fuel is to produce ATP. ATP is used to allow the muscle to shorten, allow the proteins to work. Well, that fuel has to come from somewhere, right? Well, it comes from glucose and oxygen. The two are going to be used. Glucose will be broken apart, and then the bonds in glucose will be used to make ATP. So glucose and oxygen is our fuel source. Well, in skeletal muscles, glycogen is stores of glucose. Oxygen stored in the myoglobin. So what we see in skeletal muscles is we have oxygen already available. It's already going to be bound with myoglobin, red-colored oxygen-binding protein, very similar to hemoglobin. This myoglobin is what makes red meat, right? It's red because the skeletal muscles of that animal contains lots of myoglobin. What else do we see? Not only do we require myoglobin, and therefore we can store oxygen inside the skeletal muscle cell, but we can also store glucose because all skeletal muscle cells contain glycogen. Glycogen is how we store glucose. Is glucose bonded to each other into making long chains of just pure glucose. Once we make the glycogen, what's made out of glucose? When we need it, we just break it apart. As we break it apart, the individual glucose molecule will then be used along with oxygen from myoglobin. They will both be used to drive oxidative phosphorylation and the Krebs cycle and the mitochondrial electron transport. So then we produce a lot of ATP. So important thing to understand, you don't need blood flow immediately to bring new supplies in. The first 5 minutes, right, of working out, you are gonna break down the stores of glycogen, break down the stores of myoglobin. So then you can have available fuel already in the muscle. Once you got rid of it, after 5 minutes, then the muscles that you're contracting, that you're training, those muscles then require blood flow to bring fresh nutrients in. There's a reason why they always tell you when you're doing aerobic exercise, the goal is to do it for more than 30 minutes. Usually after the first 5 minutes, then you start breaking apart, right? And you get rid of the glycogen. And then by the end of 20 some minutes, there's very little glucose and very little oxygen in the cell. So then you require blood flow. So then you can bring oxygen to that area and you start getting rid of fats and fatty acids. So then you start burning fats after 30 minutes. Perfect. Now, next, oops. All right, next, if I can get this stupid thing to work. All right, next we're gonna do is, all right, I'm gonna see if I have to go full screen on this thing. All right, I think I'm gonna have to go full screen again. Right. Next, we're gonna look at this, all right? Again, typical cell does not have, so a typical cell does not have myoglobin. Typical cell does not have these myofibrils. They don't store glycogen. So you've heard of kids eating pasta before a game. right? The main purpose is to build up your glycogen stores. In reality, it doesn't work great because only skeletal muscles and the liver can store glucose as glycogen. So most kids, when they eat pasta for the pasta dinners that before a meal or before a game, what happens is they overeat it, and there's only so much storage of glycogen we can have. After you go beyond that, you start turning that glucose, the pasta, the sugars in pasta, you start turning it into fat. So then you can store more of it. And there's almost no limit to how much fat we can store. Now the T-tubules, I'm just gonna come back here. The T-tubules are these right here. And the transverse tubules, again, are invaginations of the plasma membrane internally. So then if you have an action potential spreading throughout the length of the plasma membrane, the sarcolemma, action potentials triggered throughout the sarcolemma, then the action potentials are going to go down these tunnels. As it goes down these tunnels, again, right, these tunnels are part of a triad system. The tunnel is a transverse tubule. The triad system contains calcium storing terminal cisterns on each side. And what we're going to see is at rest, the sarcoplasm is going to have incredibly low amounts of calcium. Calcium is the most important key to how a muscle gets stimulated and then contracts. It is incredibly important to have calcium, right? And what we do is at rest, calcium is stored and concentrated into these terminal cisterns on each side right here, right? Inside the sarcoplasm, because the calcium has been removed, the sarcoplasm has incredibly small amounts of calcium. That low calcium environment is going to be very important, especially when we want the muscle to rest. Why is there such a low amount of calcium in a skeletal muscle cell at rest? Because all the calcium is stored and concentrated on dilated sacs called terminal cisterns. Now, on a regular cell, we see endoplasmic reticulum, smooth, and we see rough endoplasmic reticulum. In a muscle cell, we also see rough endoplasmic reticulum, right? Because we need to make lots of the proteins. But the smooth endoplasmic reticulum, a lot of it have been modified to form this network, this web-like network that surrounds each myofibril. That is called the sarcoplasmic reticulum. The sarcoplasmic reticulum allows us to carry calcium from the sarcoplasm and then concentrate it in these terminal cisterns. The terminal cisterns then surround the transverse tubules. We're going to see once an action potential gets triggered, an action potential then spreads through the sarcolemma. As it spreads it through the sarcolemma, it goes into the transverse tubules. As the action potential goes down the transverse tubules, it's a signal, right? Because now you have an action potential down the transverse tubules, it triggers the release of calcium from these from receptors found in this terminal cistern. So then the terminal cisterns that were holding the calcium will release the calcium into the sarcoplasm, which will then allow muscles to contract. So the whole purpose of the transverse tubule system is to carry action potentials interiorly, which will then causes the release of calcium from the terminal cisterns. Perfect. Right? Again, this kind of a couple situations I want everyone to think about at rest. At rest, sarcoplasm, low calcium, incredibly low calcium. That is what allows the muscles to rest. And anyone, all right, at rest, large amounts of glycogen. At rest, large amount of oxygen stored as myoglobin. Again, review this for the transverse tubules. Again, tunnels connected to the plasma membrane. If the plasma membrane, the sarcolemma, depolarizes and an action potential spreads throughout, it's going to go then down these tunnels, and the tunnels are going to carry that action potential deep into the cell, where as it goes deep into the cell, it's surrounded by the terminal cisterns. The terminal cisterns store calcium. They release the calcium when the transverse tubules start to depolarize. Now, the mitochondria lies in rows throughout the cell. Makes sense why we have so much mitochondria here. We have so many mitochondria because we need so many ATPs for muscle contraction to occur. And what we're going to see is ATP is fuel, right? It's like our battery. So what happens when you contract the muscle actively? It makes sense. You need fuel. You need to break apart ATP and drive the head of the myosin to start to move, right? The weird thing is you need ATP not just to contract, but you also need fresh ATP to relax. And we'll talk about why in a minute. Again, right, myofibrils, good picture right here, kind of blown up, make up, and you can see lots of myofibrils make up the interior compartments of the cell. There's so many of them, it pushes all the other organelles to the side. The myofibrils separated from each other by the web-like encircling of the sarcoplasmic reticulum. The sarcoplasmic reticulum then ends as dilated sacs called the terminal cisterns. So this sarcoplasmic reticulum, when it gets stimulated, there's a direct release of calcium. But the direct release of calcium, right, the calcium be released because the action potential spreads on the transverse tubules here. As you stimulate this area, you'll release calcium only in this area. The area back here, or in front right here, well, the action potential hasn't spread there yet. You're not going to release calcium over here, right? Action potentials in muscles are very similar to what we see action potentials in an unmyelinated nerve, meaning that each segment has to activate the next segment, right? And it takes a while. So let's assume this is, right, a little bit, you know, this is the area we stimulate, right? As we stimulate this area, action potential goes down the transverse tubules all the way throughout this segment and all these transverse tubules. That triggers the release of calcium only from these sarcoplasmic reticulum right here. So then only this area over here and over here, only these area will have the release of calcium, and only that area will contract. That's going to be important coming up. So again, sarcoplasmic reticulum encircles each myofibril, similar to the smooth endoplasmic reticulum in non-muscle cells. And in the relaxed muscle, it stores calcium and concentrates it in the terminal cisterns. So I'm just going to write that up right now, so then we can understand that it's in the terminal cisterns that we really store the calcium. It's not just we store it everywhere throughout the system. No, it's only in the terminal cisterns. So that's where we store it. And we're going to see again, I've mentioned calcium a lot. It's incredibly important for muscle contractions, for good muscle contractions. Now next, we're going to see, why can't I move this slide? Here we go. All right, next we're going to talk about, and here like with, you know, the connective tissue coverings of muscles, right? The epimysium, connective tissue that surrounds the whole muscle. Perimysium, right? Now epimysium right here is going to surround the whole muscle group. it becomes continuous with the tendon. So with the epimysium, your bicep muscle is going to have epimysium covering the whole cluster of that, all those fascicles kind of embedded with each other. This is going to sound gross, but if you've ever cooked beef tenderloin, if you look at it real closely, has what looks like, some people call it silver skin on it, right? That's that fascia that we call the epimysium. It covers the whole skeletal muscle. That's a beef tenderloin. When you eat steak, when you eat beef tenderloin, what are we eating? We're eating muscle. I know, sounds gross, but that's what we're eating. We're eating the cow's muscle. And a good butcher will actually tell you the actual muscle you're eating, which even for me can be a little too much, right? I don't even need to know the exact muscle that I'm eating of a cow. It becomes a little too real that it's a cow. It's an animal that we're eating. So what happens is that when you do cook though, you will see fascia. You'll see what looks like little thin, and it looks silvery connective tissue covering the whole external part of the muscle. That's the epimysium. The perimysium is only seen when we cut it in half and then we look under a microscope. Under the microscope, this perimysium, hopefully you can see it right here, right? This perimysium are found, and it connects each cluster of skeletal muscles that are arranged in fascicles. So this perimysium kind of winds around each of the fascicles, keeping these fascicles nice and taut and connected to each other. And again, right, a fascicle, clusters of skeletal muscle cells grouped together. As they're grouped together, it's surrounded by an external covering called the perimysium. So perimysium covers and surrounds each fascicle. So you see perimysium here, around this fascicle, another perimysium around the next fascicle, another perimysium around this one, and around all of these. That's a perimysium, right? The endomysium, right, is going to connect one skeletal muscle cell to the next inside each of the fascicle, right? Now, endomysium right here, and you can take a look, and this is very important to understand as well, right? Simple thing is this. We think that when we bring blood, we're bringing nutrients to the cells. And keep in mind, blood never mixes. It never directly touches any of the target tissues that it's going to supply. So if we see the skeletal muscle cell right here, what we're going to see is there's always a blood vessel in between the blood itself coursing through and the target tissue. Blood never drains into the target tissue, in other words, right? Now, in order to have gas exchange, nutrient and waste exchange, we're going to bring blood to smaller capillaries. The capillaries are going to be thinly walled. That allows diffusion of gases. But there's never any direct mixing of red blood cells with a skeletal muscle cell, with a neuron, with any target tissue. There's always a blood vessel in the way. And here you can see how the fascia right here is connected to the epimysium. Now, the muscular fascia, dense fibrous connective tissue, has no give. Sometimes when you damage a cell through trauma, you can have something called compartment syndrome. Compartment syndrome then means that you have trauma over here and there's no give in the muscle. Have you ever gotten an infection in your arm or in your fingers and it starts to swell up from the inside, right? And it feels like there's so much pressure that it can burst through the skin. Well, that's what compartment syndrome is. You've damaged the muscle somehow, and there's causing leakage then of the capillaries, and sometimes even some of the main arteries supplying that muscle. as the blood starts to pool into the muscle cell, well, the fascia has no give. So it creates a lot of pressure in the muscle. That pressure in the muscle then stops any further blood flow. If we don't treat it, the muscle will die because there's no more blood flow. If we do treat it, you'll have this, right? When we do treat it, and hopefully we do, you will have something called compartment syndrome, and we need to alleviate it. And it's pretty ugly, so just a little warning. Whoops. So here's compartment syndrome right here. And you'll see with compartment syndrome that in order to actually relieve it, you actually have to cut through the fascia, cut into the skin, through the skin and the fascia. If you don't, you're going to lose that arm or leg, right? Usually there's time, but take a look at all these different areas that we can have it. In the calves, right? Look at that person right here. All right, so they had compartment syndrome over here, and in order to relieve that pressure and prevent an amputation, they had to cut through the fascia. As that cut through the fascia, what happens? The muscle inside is so swollen that usually it bursts, right? Now, at that point, there's no way we can bring the actual skin back together to heal. And we wouldn't want to do it anyways, right? But what we do is do some kind of retention sutures so then it doesn't completely move away from each other. Now, even at the best situation, you'll have this kind of look to it where they have to cut through the fascia. At the worst situation, and you don't get it treated in time, you'll have what happened to this guy. Miles O'Brien was well, he still is, right? Journalist. And with this journalist, he was somewhere on, right, for work. And if you take a look right here, he's missing an arm, right? What happened with Miles O'Brien is he had damage in his arm from reporting on a story, and then a camera bag hit him in the arm. That's just a little camera bag, but he didn't get it treated. And instead he went home to his, I believe he went on vacation after he covered that story, and he didn't want to leave his vacation and his family. Eventually it got so bad that he had to go to the ER. And by that time it was too late to decompress the compartment syndrome, and they had to amputate his arm. So compartment syndrome is usually pretty violent, and it's a pretty ugly situation to have. The only good news is that it's pretty rare. So that is what can happen. Amazingly, it doesn't happen as much as it possibly could, which is good. Next, right? Now, what do we need to know here? Number one, what we mentioned before. All skeletal muscles require motor neurons to be stimulated. Now, we will see cardiac muscles, smooth muscles, doesn't necessarily need motor neurons. It can get stimulated by motor neurons, but there are different other ways to activate those muscles. In terms of skeletal muscle, the only way to activate a skeletal muscle is with a motor neuron, a neuron that is specific to cause muscle contraction. Once a motor neuron is going to fire and get stimulated, then the motor neuron is going to stimulate the muscle fibers to contract. Nerve cells with cell bodies in the brain or the spinal cord, and axons that leaves the spinal cord and goes to the target muscle. As it leaves the spinal cord, it reacts with other and gets bunched and grouped together with other axons to form part of the nerve, right? Nerves are not like, if we're talking about your biceps, the main nerve that stimulates the biceps is called the musculocutaneous nerve. Don't need to know that for right now, but just understand, we know the name of the nerves. The musculocutaneous nerve is going to have a mixture of motor neurons that goes to different cells of the biceps and sensory neurons, right? So nerves are mixed. One giving motor signals to the muscles of that area. Then you have motor sensory neurons receiving sensory innervation from that area as well. The motor neuron's main purpose is to stimulate the skeletal muscles that it's connected to. The contact point between the neuron and the muscle is called the neuromuscular junction. That's where we're going to have the nerve stimulate the muscle. Skeletal muscles always require motor neuron activation. And not only that, the skeletal muscles will atrophy. It will atrophy if no motor neuron activity. Meaning what? People that have spinal cord injury. When you have spinal cord injury, the spinal cord has been kind of damaged, and the motor neurons coming out will also be damaged. And because it's damaged, it's no longer sending action potentials to the muscle. The muscle now can't work, can't concentrate, and with a lack of activation, it actually atrophies. So every now and then you'll watch videos where people that have paraplegia, quadriplegia, having lots of physical therapy, lots of occupational therapy, even though they can't move that limb. The main purpose is to keep that muscle from atrophying. Oops. Right. So this kind of reviews what I mentioned, like some of the differences between a normal cell and the skeletal muscle cell. Right. One of them was at Sarcoplasmic Reticulum, only found in muscle cells. Sarcoplasmic Reticulum stores calcium in its terminal cistern. So I'm gonna just write this in. Stores it here in the terminal cistern. And then as it stores it, this terminal cistern will release that calcium when an action potential gets triggered through the transverse tubules that are part of the triad system. All right, now in terms of the proteins right here, we're gonna see thick and thin filaments. The meeting point or lack of overlap between thick and thin filaments creates the banding. There's the light I bands, anisotropic, and then you have the darker A bands, anisotropic, right? Now, how can you tell the bands from each other? Simple, right? I bands are light. There's a I in light. I know, very, 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 right? Very simple, right? Dark bands are the A bands because there's an A in it. Simple ways to help you memorize which is which, right? What else? The I bands are regions that only contain thin filaments. In the overlap area between thick and thin filament, then we see the A band, all right? A bands. So overlap between proteins creates the A bands. Now, why do we see this light-dark combination? Keep in mind that when we're looking at a microscope, light is coming from underneath, right? It's going to try to go through the skeletal muscle cell. So that as light goes through the skeletal muscle cell, let's see if we have a picture of it right here. Right here, right? So here's a light microscope, and muscle is, actually I believe that might be an electron microscope. So even that's very similar, right? So what we're gonna see is the more dense areas right here. Well, the dense area is going to contain thick and thin filaments. So it makes sense that there's more protein there and there's more density there. Light has a harder time going through that region, right? Because light has a harder time going through that region, it appears darker. That's this area right here. That's our A band right here. Right? The I band is a region that is void of the thick filament. Right? The I band is just a thin filament then. Well, thin filament is thin. So if it's thin, more light can go through. Right? If more light can go through, it appears lighter. And that's what we see here. The lighter I bands is only the thin filament. And now you have the Z disc that bisects the actual the I band right here. Perfect. All right. Now, what else do we see? In this area right here, so we see that in the A band region, that there's a little region where it's a little lighter in color in between. See that? This whole thing is an A band, but then you see this region right here that looks a little bit lighter. What is that region? Well, that's the region over here. That's the region where it's only thick filament. There's no overlap of the thin. The thin stops over here on each side, right? All we have then are the thick filaments. And because it's only one of the proteins, it's going to appear, right, a little bit lighter in color again. But then again, if you take a look at this and compare it to this H zone region still looks a little bit darker than the I bands. Right? So the H zone region is a region where there are no actin myofilaments. And then, right, we're going to see that when we contract the muscle, myosin's head is going to pull on actin to the middle. Myosin's head is going to pull on actin to the middle. What happens when you pull everything towards the middle? Everything shortens, right? As things shorten, this H zone is going to start becoming smaller and smaller. By the time you have a fully contracted muscle, the H zone should disappear because you're moving the thin filaments, all right, always to the middle. So once you have fully contracted muscle, the thin filament is going to be right here instead of over here. The thin filament is going to be over here instead of back here where it started. Now that H zone is going to be very small. It can also be obliterated, right, with strongest contractions. All right, let's talk about thick and thin filaments, and then we'll take a little break. Right, so actin and myosin are the main players here. If you take a look, you'll have a Z disc, and we'll talk about it in a second. The functional unit of a skeletal muscle. So we have a pretty long skeletal muscle cell, right? It's going to be broken down into functional grouping or units called sarcomeres. So the sarcomere is the space, the span between one Z disc to the next. The Z disc is the anchoring point for our actin, thin filament, and our myosin. It allows to have structure. So then actin and myosin can interact, actin can move, and then there's something that anchors actin in place. Perfect. Now, here's one sarcomere over here. They color code this. So then the purple actin, that's what we see here, purple, and the green myosin, right? So in some ways, it's good they color code it. I wish it color coded like a little bit better color that matches our muscle cells, but purple and green makes it easier to see. So let's talk about these muscles and these myofilaments found in the muscles. So again, you have a skeletal muscle fiber or cell inside multiple myofibrils. Now we're looking at one of those myofibrils and discussing the proteins that are found inside one of the myofibrils. You're gonna have a lot more of them. All we need to do is look at one. right? When we look at myofibrils, it's made out of functional proteins that allows for movement. So we have actin is the first one we'll discuss. Actin, 2 strands of fibrous actin forms a double helix structure, extending the length of the myofilament, attached at either end of the sarcomere. Right? So let's take a look again at that picture. So here's the actin that looks like little grapes on top. It's going to be attached and anchored on each side to a Z disc. The Z disc provides structural support, and we're going to see with the Z disc, it allows what happens to the actin to move the actual cell. All right, we'll talk about what that means in a second. Now, with actin, you see F-actin and G-actin. The G-actin is the areas or the actin that contains these yellow little pits right here. Those pits are the binding sites for myosin. When we contract the muscle shortens. We're going to see the muscle itself, we don't get rid of any of the proteins and shorten the proteins. We're going to see that as we shorten and contract, myosin is going to pull on actin on each side, and they're going to pull everything to the middle. As you pull everything to the middle, the cell's going to shorten. Exactly what a muscle contraction is. Now, we'll also see that the myosin is a thick filament, right? And it contains these heads that pop up. Myosin needs to be able to interact with actin. When it does so, it forms a cross bridge. So a cross bridge is the interaction, the meeting of myosin's head with the binding site on actin. All right, so usually, besides these two functional proteins, we'll see other regulatory proteins that regulates when we can interact. The last thing we want is to have our muscles contract when we're not expecting it. Think of a muscle cramp, right? I've never heard of somebody enjoying a muscle cramp. So especially if it's like, 3 a.m. in the morning and you're having a muscle cramp, which is usually when they happen. Now, to ensure that we don't have that unwanted accidental contraction, at rest, we have 2 main proteins that regulates the interaction between actin and myosin. The first one is called tropomyosin. Tropomyosin, and I want you to understand this analogy, tropomyosin is the gate, right? You go to watch the Bucks game, right? You go to, I don't know, right? You go to watch a Packer game. You go to watch a baseball game. And as you go in, there's always a gate, especially with a Bucks game. You're parking in a structure. As you go in, there's a gate that stops you from going in without paying. Once you pay, the gate goes up, right? Well, the gate is tropomyosin. Tropomyosin stops and blocks the binding sites. So at rest, you have tropomyosin in the way, and actin and myosin cannot make contact and form cross bridges. Perfect. That stops unwanted contraction because tropomyosin blocks the interaction between the 2 functional proteins. Well, what happens if we want to contract? What happens when we're trying to get in and now we're going to pay? Well, we need a gatekeeper. We need some kind of machine so then I can put my credit card in that lifts the gate. Well, in our body, it's not a machine. It's a protein, right? We have a gate, and now we need a gatekeeper. That's where troponin comes in, right? Troponin right here is a gatekeeper. What it's going to do is this. It's going to be a calcium binding protein on one side. On the other side, it's attached to tropomyosin. When calcium is available, it binds to troponin in red here. Then tropomyosin is moved out of the way because as calcium binds to troponin, it causes a change in shape of troponin. Troponin then shifts tropomyosin out of the way. Doesn't destroy it. All it's doing is lifting the gate. That's it. As you're lifting the gate, actin and myosin can now form cross bridges, interact, and now you can have a muscle contraction. Here it is right here, right? One region is bound to the actin, another binds to calcium, and then the other long region then moves tropomyosin out of the way. So this tropomyosin and troponin, those are our regulatory proteins that regulates when the other two proteins, actin and myosin, can interact. All right, we talk about it here, so review it when you have a chance. Let me talk about myosin, and then we should be done. All right, myosin's head right here is the important aspect of it. I forgot, it's already like 1:20. So we just keep going, and right, for our 12:20. We'll just keep going for another 15 minutes or so. All right, and then we'll finish. So myosin is also called the thick filament. Now, myosin is the main mover here. Actin doesn't really, it's what's getting moved, right? Myosin is providing the energy for movement. So myosin, many elongated myosin molecules with heads shaped like golf clubs. The molecules consist of myosin heavy chain wound together to form rod portion, the handle of the golf club, and exits out as myosin heads. Right? Now here, when we're looking at it, it only looks like there's an up and down facing myosin head. Remember, when we're looking at this, this is 360 degrees though, right? So we're gonna have myosin, all right? So you're gonna have actin on top and on the bottom. Then you have actin on the sides. Then actin on the top and to the side. So there's about 6 different actin molecules completely surrounding the myosin in a 360 degree array. So then you have lots of these actins all the way surrounding the myosin head. The myosin head then faces upwards and downwards, faces laterally on each side, faces up and out, right? In other words, it's not just 2 myosin heads. That's a big thing I want you to understand, right? These myosin heads, even though they show it as just up and down, the myosin heads can face laterally. It can face downwards and lateral at a diagonal, right, as well. So then you maximize the number of cross bridges between myosin and actin that surrounds it. The myosin head has a binding site to actin, allowing it to form cross bridges. Attached to a rod portion by a hinge region that can bend and straighten. So it can move, in other words, this hinge region right here. And then to power that movement, we have an enzyme, ATPase enzyme, that breaks down ATP, releasing the energy and cocking the head of the myosin. What we are going to see is before the muscle actually contracts, right? The myosin head is already cocked, ready to go. So everything is ready to go. Myosin's head's already used the energy in ATP. The head's already cocked, ready to go. The thing is, at rest, tropomyosin's in the way. All we need is to move it out of the way. And that's where troponin comes in. That's where calcium comes in. Right? Now, this kind of goes into a little bit more detail about the Z-disc, anchors the sarcomere into the sarcolemma, and then tighten, right? Tighten elastic chains of amino acids, make cells extensible, and elastic. It's a main structural protein, and usually we're gonna see is when we contract the muscle and we measure the time it takes to contract, right? There's always a slight delay between when we stimulate the muscle and when the muscle actually shortens with the most force. That slight delay, right, number one, is due to calcium that needs to be released, calcium that needs to bind to troponin, then move tropomyosin out of the way. It's also because there's a little bit of slack due to tighten, right? You need to get rid of that slack for the muscle to contract with strength. And here's what Titan looks like. So here's Titan right here. See that? Titan kind of looks like a spring. Kind of makes sense what it does then, right? Right. Here's that look, whoops, right here, of just actin. So here's a myofibril. And what we're looking at, right, is just one myofibril in one area of the sarcomere. So this is one sarcomere right here. What we're doing is taking a cut of it and looking only at actin. So you can see there's a lot more actins than myosins. So there's this 1, 2, 3, 4, 5, 6. Right? There's 6 actin molecules surrounding one thick filament made out of myosin. The myosin heads, though, move out in this kind of array right here. So then when it contracts, it can reach up and you maximize the cross bridges. Perfect. Right? Sliding filament theory is what we've been mentioning, right? Actin myofilaments slide over myosin to shorten the sarcomere. The actin and myosin never changes length. The overlap between actin and myosin, it is what changes. The H zone. At rest, the H zone is a space between one actin molecule to the next. Right? At rest, the H zone is pretty large. When we start to contract, we're moving the actin. And as we move the actin medially, the H zone starts to decrease in size. Right? Why? Because we're pulling the actin towards the middle. The overlap between the actin and myosin becomes greater. And then the H zone starts to disappear. That is a muscle contraction. This is the sliding filament theory. Right. Now, skeletal muscles, important thing to understand is that they're in a cluster of cells that are slightly polarized. Now, when we look at skeletal muscle physiology, right, like I mentioned, this chapter is hard because most of us have never heard of action potentials or understood it, even if you've heard of it. So we introduce that concept of action potentials, and at the same time, we talk about how the proteins interact. Two different, yet both individually, hard concepts. So when you put them together into one chapter, it makes it a little bit more difficult. So let's talk about skeletal muscle physiology and how it compares to a neuron. Right? It's very similar. Now, in the skeletal muscle cell, on the very inside of the plasma membrane, not throughout the whole cell, but on the inner lining of the plasma membrane, it's slightly less, right? It has a slightly negative charge compared to the outside. Now, the outside is an extracellular fluid space. Now, if we measure the extra fluid space a little bit further away from the cell, way over here, well, there's no charge difference. The charge difference are only found very close to the plasma membrane. So very close to the outside of the plasma membrane, slightly positive compared to the inside. Again, the inside is not the whole internal intracellular fluid space. It is that area immediately close to the plasma membrane. Only that region is slightly negative. It's slightly negative at minus 85 to minus 90 millivolts. Depending on different books, they'll talk about each one. All right? At rest, then, this lining right here of the plasma membrane is slightly negative. And there's a difference in charge between the outside of the cell and the inside. Because there's a difference in charge, we say that the cell is polarized. Neurons also have that same slight negativity along the inner margin of the plasma membrane. Gives them also a slight negative charge. They're slightly even less negative. So in the neuron, the resting membrane potential is at minus 75. Now, on this I think I'm missing a slide on dystrophin. So I'm going to add it on to us, all right? Now, with skeletal muscle fibers, the resting membrane potential of minus 85 to minus 90 is due to accumulation of large protein molecules, is due to also, right, more potassium in the inside and outside. Now, what we're going to see is simple. I don't know if it's simple, but it's easy to see, right? We're going to have the sodium-potassium pump. Sodium-potassium pump pumps out 3 sodiums from the inside of the cell, and you pump it out. 3 sodiums, 3 positive charges. At the same time, only 2 potassiums are brought in. So you are losing a positive charge with each pump. If you lose a positive charge, you don't become 0, but the cells losing that positive charge becomes negative. Not only do we not, there's an unequal, right, movement of ions, so then you're only bringing 2 positive charges in, but some of the potassium that moves in, some of it leaks back out through potassium leakage channels. And we know there are a lot more potassium leakage channels compared to sodium leakage channels, right? So potassium leakage channels allow potassium to go from high concentrations inside the cell. Why do we have such high concentrations inside the cell? Because sodium potassium pump pumps in potassium. If we keep pumping in potassium, I'm going to have a big concentration gradient for the potassium. In the purple, there's a lot of potassium on the inside, very little on the outside. Big concentration gradient means potassium, when it can, will prefer to leave or efflux from the inside out. We have leakage channels that are always open, and they allow potassium to leak out. Now, why is this important? Not only do we lose that positive charge when we pump out 3 sodiums and we bring in 2 potassiums, but some of the potassiums leak back out, losing even more positive charge. That helps account for a lot of the negativity. Perfect. Right? It also accounts for the sodium and potassium concentration gradients. High sodium on the outside. Why? Because we keep pumping it out. Low sodium on the inside because we keep pumping it out. Opposite's true for potassium, right? Since we keep pumping it in, high potassium on the inside, low on the outside. It's going to be allowing what the movement of those ions are going to allow for rapid depolarization and repolarization, right? Now, ligand-gated channels are the ion channels. We'll talk a little bit about action potentials and then we'll stop. So in terms of ion channels, we have here 2 main types we're going to be very, very concentrating on. Number 1, ligand-gated ion channels. What is a ligand? A ligand are any molecules that can bind to a specific set of receptors, right? So a ligand then can be anything, including a neurotransmitter, norepinephrine, epinephrine. They're ligands. It can be a hormone, insulin, ligand, right? So it's a large amount of chemically active molecules that can bind to its own receptor. So all of your hormones are types of ligands. All of your neurotransmitters are type of ligands. So ligand is a very general name. One of the main ones we're going to talk about is a class called neurotransmitters and a specific neurotransmitter called acetylcholine. Acetylcholine binds to its acetylcholine receptors, right? In the process, the acetylcholine receptor is a sodium channel. At rest, when there's no acetylcholine, the sodium channel is closed. But when there is acetylcholine release, acetylcholine binds to its receptor, opening up sodium channels. Where does sodium do? All right, what does sodium do? Sodium then moves into the cell because of the concentration gradient, allowing us to slightly depolarize, then eventually hit threshold. At threshold, a second channel opens up. These are called voltage-gated channels. So we have ligand-gated that starts the process of an action potential. Once sodium goes in and you hit threshold, then the voltage-gated channel opens, specifically sodium voltage-gated channels. The thing about these voltage-gated channels is at rest, we're at minus 90, minus 85. Well, you need a threshold voltage, right, to get to for these to open. And the threshold is minus 55 millivolts. Somehow, we need enough sodium to rush in. Let me just move this here so you can see what I mean. Right? So at rest, high sodium on the outside, high potassium on the inside. When I have acetylcholine bind, right? Acetylcholine binding will cause acetylcholine receptors to open up. Sodium goes in. As sodium goes in, well, what happens when sodium goes in? When sodium goes in, right, we're going to say, here's sodium going in. As sodium goes in, sodium's positive charge. So we go from minus 85 as sodium goes in to minus 75. As more sodium goes in, you hit minus 55. When enough sodium goes in that you hit minus 55, then the voltage-gated sodium channels open. As voltage-gated sodium channels open, this movement of sodium internally through these channels is what sparks an action potential, is what creates action potential, right? Then potassium channels open at 0 millivolts, allowing us to recover and repolarize. So then we can get ready for the next contraction phase. So the first time, right, we kind of introduce that thought. Here, then I'll stop after this, I promise. So here, action potential, right? At rest, we're at minus 85 millivolts, right? Let me just close that part out. At rest, we're at minus 85, minus 90, all right? And what happens is that's our resting membrane potential. Then you have this kind of slight movement, this slight depolarization. This slight depolarization, if you have like to make slides and review it physically, right? I want you to write down right here, this slight depolarization phase. And I'm gonna write it in right here, is due to sodium influx through ACH channels. So this slow depolarization is caused not by any voltage-gated channels. No, they're still closed. This slight depolarization is due to sodium going into the cell membrane through acetylcholine receptors. As acetylcholine receptors open up, sodium rushes in, and then eventually, if enough sodium rushes in, sodium's positive charge. So you're adding positive charge. As you add positive charge to something negative, right, you're making it less negative. So now as sodium goes in, you go from minus 85 to minus 70. As a little bit more sodium goes in, you go to minus 60. And then if enough sodium goes in, usually there is enough, you hit threshold. At threshold then, voltage-gated sodium channels open. That happens at threshold voltage, right? And threshold voltage always is at minus 55. Now, right, if you don't hit minus 55 millivolts, you hit minus 56 millivolts and go back down. Well, you don't hit threshold, you don't open the voltage-gated sodium channels. You don't have any muscle contraction at all. Not a twitch, nothing, right? This is all or none. So if you hit threshold, then you'll at least have a twitch contraction. If you don't hit threshold, you go all the way back down. Now, if you do hit threshold, voltage-gated sodium channels open, and now more sodium rushes through. And you have the depolarization phase. Perfect. Now we're stimulating that segment. Then that segment will allow the other segment to be stimulated, like a domino, right? Like those line of dominoes. When one domino goes down, it causes the next domino to go down. Same thing here. As one segment of the plasma membrane depolarizes, it creates sodium influx to the next segment, causing that next segment to hit threshold and depolarize. As that next segment depolarizes, sodium rushes in through the voltage-gated channels. Sodium then causes the next segment to hit threshold. Each segment always causes the depolarization of the next. That depolarization phase is then followed by a repolarization phase where voltage-gated potassium channels open. As voltage-gated potassium channels open, potassium leaves the cell, making my cell more negative and allowing my cell to recover. So then I can get ready for the next cycle of contraction. So repolarization due to opening of voltage-gated potassium channels and potassium efflux. Now, at this point, right here, right, at about positive 30, my sodium, voltage-gated sodium channels become inactive. They don't totally shut down and recover yet. They just become inactive. And throughout repolarization, the voltage-gated sodium channels recover, and they move from being inactive to be fully closed. Once the voltage-gated channels are fully closed, now you can reopen it. And that's what we see down here, right? Once we go all the way back down to resting membrane potential, we can now re-stimulate that segment of the plasma membrane. Perfect. Again, right, this kind of shows it again. Now, the one thing I wish they did is kind of show how acetylcholine receptors work right here, but we have other slides later on that shows it, right? So right here, at rest, outside the cell, slightly positive, inside slightly negative. High sodium, low sodium. High potassium, low potassium. What happens, right? What happens, right, is that this is what we see at rest. When we depolarize and we hit threshold, voltage-gated sodium channels open. Sodium rushes into the cell, and when sodium rushes in, the thin region right next to the membrane, well, sodium's rushing in. It's positive charge. You become positively charged, right, due to sodium influx. Because you're losing sodium right here, the very near extracellular fluid space close to the plasma membrane starts becoming negative. So there's an inversion of charge. This area right here is where the actual cell depolarizes and starts to activate. And then once you've activated that region, voltage-gated sodium channels become inactive. Voltage-gated potassium channels open up, and potassium leaves the cell. As potassium leaves the cell, you lose positive charge. The area that receives a positive charge becomes positive. The area that loses the positive charge becomes negative. Now, we are becoming more negative and recovered and repolarized, which is perfect, right? We need to always depolarize and then always repolarize. That allows us to stimulate and then recover. So then we can stimulate and recover again, right? And again, the last thing I'll talk about is here, right? Each segment always causes the activation of the next segment. That's what I want you to see here. What happens is this. When this region of the plasma membrane, of the sarcolemma, depolarizes, voltage-gated sodium channels open up on both sides right here. Sodium rushes in to the cell, causing the cell to become positively charged or depolarized. As sodium rushes in, this region in front right here hasn't undergone an action potential yet because this is the original starting point. So what happens? Well, this region right here is at rest. with a resting membrane potential of minus 85. The sodium coming in here doesn't just sit still. The sodium moves forward. As it moves forward, you're adding positive charge moving forward, causing this region to hit threshold. As this region hits threshold, voltage-gated sodium channels open up in that region. Sodium comes in the next region, right? Causing this region to become stimulated now. As sodium comes in, sodium goes forward, and sodium goes backward. As sodium goes backward, well, that region is in recovery. It's in the repolarization phase. So even though sodium moves backwards here, it can't re-stimulate going backwards. Sodium going forward encounters a region that is yet to activate. So it's ready to go. As sodium goes forward, it causes the next segment to have an increase in charge because sodium is positive charge, right? As it moves to the front here, that positive charge increases the resting membrane potential from minus 85. Now sodium goes in that segment. Minus 70, more sodium comes in, minus 55. You hit threshold, now voltage-gated sodium channels open up here. Now sodium comes in, and the whole thing on this slide is that each segment always causes the next segment to depolarize. And the reason for it is because when sodium comes in, right, that sodium movement, sodium influx causes depolarization, but it doesn't just sit in the segment. It's going to move forward and it's going to move back. When it moves forward, it encounters a membrane that is yet to depolarize. It's at rest. So when sodium moves forward right here, sodium is positive charge. It will quickly build up charge. So then you hit threshold in the next segment. As you hit threshold in this next segment, this segment will be the next to depolarize. Then this one after it. And then this one after that. So there's a sequential movement of that action potential. That's why we don't usually like calling it an electrical impulse. Because an electrical impulse, you think that it's just like an electrical current. It's just moving. In reality, there's a slight delay. This area gets stimulated, sodium rushes into the next region, and there's a little delay until the next region stimulates. And then there's a little delay until the next region stimulates. Eventually, though, when we look at it at its totality, then we see that it does move from segment to segment to segment to segment, all the way down the length of the muscle. Eventually, the action potential spreads into the transverse tubule, and that triggers calcium release. Now, the big thing is here, understanding that the sole purpose of the action potential is to release calcium, is to signal the release of calcium from the sarcoplasmic reticulum. That's the sole purpose of the action potential, is to signal the sarcoplasmic reticulum to release calcium. Right. I will end right here.